Announcements. All right, I'll turn it over to Bree, who will introduce our speaker today.
Hi everyone, I'm so excited to introduce Alex. This is Alex, which is a very rare photo because it's one of the only photos ever taken of Alex where he is not holding a snake. And between then and now, not much has changed except less overalls, more plaid, and more snakes. So between then and now, Alex has traveled through many parts of the world collecting many different kinds of amphibians and reptiles. It's his true passion. Along the way, he's intersected with many of the greats that have also come through here. He worked with Harry Green as an undergrad, and when he graduated from Oberlin in 2010, he made a very good decision, which was to come to White Sands for the summer, and that's how Alex and I met and began a collaboration that has lasted the better part of this decade. So I just wanted to make a few somewhat unplanned remarks about Alex before he starts his talk. Alex is an incredible herp enthusiast. So as most of you know, this is what he loves, and it's kind of like the endless summer, except of herp trips. And I did want to add one little warning, because Alex is constantly trying to rally people to go out, and it's a great idea most of the time. But this was a great warning, which I'll read because it might be too small. This is a quote from an anonymous person. "We all have stories of Alex's intense excitement anytime he sees a herp and how he immediately runs off. In Panama, he stabbed me in the leg with his snake hook when he started running after getting excited about seeing a snake." So we have very little evidence that Alex has actually spent any time indoors during his PhD. This was the only photo that anyone sent me of Alex inside, although it was looking at a snake or a lizard fossil. And what has been impressive to me about Alex during his time in his PhD is that he's really done a PhD on his own terms. Alex is an incredibly deeply thoughtful person, which you can see as evidenced in these photographs. And he is courageous in his willingness to change course if he feels like it's the right thing for himself. So he came in really deeply interested in convergent evolution, and he'll share some of that work with you. But partway through his dissertation, he decided that he also wanted his work to have much more direct conservation relevance, and so he actively sought out collaborations with agencies and ways to make his work really translate into on-the-ground management solutions, and so he'll also share some of that work with you today. The other theme that came out when I asked people to send slides of Alex was like, cool guy Alex, which I have to admit, like, I don't know that guy. I don't know that guy as well as I know, like, the enthusiastic herper guy. I don't know now I don't even know what I was going to say about it. I totally derailed myself. Okay, this is the Alex that I know, and this is the Alex that feels like it's almost the same as the like little overall Alex, right? Which is like that enthusiasm for the natural world, and that's something that is probably unrivaled in a student that I've met, is just like that straight up enthusiastic passion about what makes this planet so incredible and unique. And that is the face of wonder that I hope that we all keeps us all young. So in closing, thank you, Maddie. To the pun-loving guy, I want to say, as long as you, oh wait, that's not right, that as you approach, that's bad grammar, the tail end of your PhD, we're all excited to watch you snake your way to a new adventure, but we will totally miss you. And there is no better herp nut than a Krohn nut. You have to eat that while giving your talk.
I'll be short on time. Hey, it worked. Thank you very much, Bree, for that introduction. I have been coming to the seminar for nearly 5.5 years now. I've been a part of this community for that long. I've gone herping with you all. I've talked science with you all. I've made friends with a lot of you, and so I've wanted to be a part of this community for more than 10 years now, and I've been a part of it, and it's been just totally rewarding. It's been just an amazing experience and an honor to be a part of this community, and it's one that I hope to be a part of for the rest of my life. And so, to make a very long story short, thanks for having me. So I'll start with an overview of the talk that I'm going to give. It's not going to be your typical kind of 40-minute talk on one subject. Instead, just like on Passover, we'll go over the 4 questions. And these are the 4 questions that motivated my dissertation research. 3 of them are on lava flow lizards, and then we're going to change gears a little bit, as Bree alluded to, and we'll talk a little bit about reptile food, voles, and each one of those little questions will have kind of a motivating factors section, and then results and many conclusions. And then we'll zoom out and take a kind of big picture look from 5.5 years in grad school. So as Bree mentioned, one of the really big motivating factors behind my research was convergent evolution. As I went out and caught reptiles and amphibians across the world, I noticed that strangely, a lot of the species looked really similar. Even though they were very distantly related and found all over the world, they had this striking similarity. That's convergent evolution. And one of my favorite examples are these 3 vipers. So this is a true viper from West Africa, a pit viper from the Philippines, and another pit viper from Costa Rica, only vaguely related to each other, but only related in the fact that they are vipers, but they've all convergently evolved this semi-arboreal yellow ecomorph in all 3 of the tropical regions. I was really interested in what factors promoted this convergent evolution. Another maybe more famous example are these 2 lizards. And so this is the thorny devil from Australia and horned lizard from the southwestern US. Again, these are not even in the same family, but they both independently evolved what I consider to be a really striking and unique ecomorph as terrestrial ant-eating lizards. That was really, really striking to me and something I wanted to study more. Lizards have been well studied in the realm of convergent evolution. You don't have to read all the details of these studies, but I'll walk you through each one by way of summary. And so probably most of you are familiar with the convergent adaptive radiations that are replicated on each of the Greater Antilles Islands in anoles. It's a really amazing example of convergent evolution in an adaptive radiation, in multiple adaptive radiations. Other folks have looked explicitly comparing the Australian desert herpetofauna with the North American herpetofauna and found that indeed the entire communities converge in both ecology and morphology across these really disparate geographical and evolutionary timescales. And then some folks have taken that a bit further. Eric Pianka and Lori Vitt have recently been trying to create kind of a periodic table of niches where they deconstruct the aspects of lizard life history, natural history, and other aspects of niche space to create a periodic table of niches whereby you can take distantly related species and kind of assess convergence by whether or not they fall into these really similarly conserved niches across the world. But as I look at this, you may notice that we're talking about convergence at the phenotypic level, but when you look at lower levels of organization, sometimes that similarity may fall apart. So I'll give an example that we're all hopefully familiar with. So bats and birds both have wings. They're convergent at the level of having wings, and for the function of and they use them for similar functions to have propelled flight. But when you look a level deeper, you can see that those wings formed from different ways. And so different bones were elongated, different bones were fused, and so that similarity kind of seems to fall apart when you look a little bit closer. Going to something a bit closer to the things that I study, all 5 of these species, really distantly related, have all independently evolved blanch coloration. We know in these 4 species it's for coloration to blend in with white sands or gypsum sands or beach sand. And mammoths were actually blanched in coloration as well, although we don't really know why. And then, again, you don't really need to know the details of this. I'll walk you through the general principle. But actually, all 5 of these species share amino acid substitutions at an important gene in the melanin synthesis pathway, MC1R. And even though they share similar mutations at the same gene, the functional consequences of those mutations vary in these taxa. Sometimes that mutation is actually responsible for decreasing the production of melanin and thus this blanched coloration, and other times it's not, and the functional consequence is a bit more ambiguous. And so again, they're convergent at this phenotypic level, but what I went back to that original question of what factors promoted convergence at the phenotypic level and then a similarity or lack thereof at these lower levels of organization. I'm definitely not the first person to ask this question. In fact, we know that these 3 things are 3 of many that affect the likelihood of convergence. So in the most general sense, the stronger the natural selection is for a certain phenotype, the more likely 2 disparate species will converge on that phenotype. Demography also modulates the likelihood of convergence, right? If we have 2 very, very large populations, there's a greater likelihood that they will independently have similar mutations in the populations. And then, as Simon Conway Morris and Stephen Jay Gould have argued for decades, or did argue for decades, history and contingency, we know, play a role in convergent evolution. And I use those terms kind of broadly to both mean the kind of evolutionary trajectory that you can take in the future depends on how on the evolutionary starting point today, and both in terms of time. We know that given enough time from bacterial and other experiments, that the longer time you give populations to evolve to some strong natural selective pressure, the greater the chance that they will converge on a similar solution to that environmental problem. So this is what I really wanted to do with my PhD. I wanted to come up with a system to look at these factors that affect the likelihood of convergent evolution. And so in order to do this, I needed to find a system where there was replicated evolution of a similar phenotype across distantly related taxa, because I didn't want just kind of a one-off episode of convergence. I wanted to kind of speak more broadly. I needed those replicates to be in a geographic area so that I could assess the role of demography on that convergent evolution. And then I needed a kind of time axis as well. I wanted to see, for example, does the time since the putative adaptation has occurred, or since this novel selective environment has existed, does that affect the probability of convergent evolution? And that's exactly what I found. So that brings me to the other, maybe more obvious motivating factor, the reptiles that I studied. These, in my humble opinion, are the most interesting reptiles on Earth. So these are just 6 of many other species that have colonized lava flows in south central New Mexico and have evolved to be darkened in coloration. And so on the left you can see the dark morph, and on the right you can see the kind of normal morph from the surrounding deserts. And I found this really, really striking. I mean, these reptiles kind of run the gamut on ecology and life history. There's nocturnal predators, cathermal predators, diurnal predators, sit and wait predators. There are active foragers. I mean, they run the gamut, and this was a really cool replication of melanism that I could study. The lava flows themselves were also a really compelling place to study this because, like I said earlier, they're within about 100 miles of each other, so they all kind of drew from a similar pool of potential colonists, and then they vary themselves in age and in size. And so I'll tell you a little bit about that now. So the Carrizosa lava flow is the youngest lava flow. It's pretty homogeneously dark in composition and is made up of pretty much sharp rocks and sharp plants. And there I studied the eastern collared lizard and the southwestern fence lizard, Crotophytus and Scoloporus. The Aden-Afton lava flow was kind of the middle-aged lava flow, and here you can see they still have really large black outcroppings, but sand has come in between the rocks and made the habitat a bit more heterogeneous. Here, Crotophytus and Scoloporus are not found, but I sampled Urosaurus, the ornate tree lizard. And finally, the Pedro Armendariz lava flow is the oldest lava flow, and it's the most heterogeneous in composition. Here, there's a lot of sand in between these dark patches, but Crotophytus and Scoloporus are also found there, and were also described as melanistic. And so that brings me to the first question, just how melanistic are these lizards? Despite this being one of the coolest systems in the world, very few people have actually studied the lizards on lava flows. I counted up that there were, I think, only 12 people that were actually first authors on papers about, that included anything on these lava flow lizards, and 2 of them are here now. Or I guess are currently affiliated with the MVZ, I should say. So rather than just go on these old faunal surveys that said, hey look, there's a dark lizard, that's melanistic, I wanted to actually go out and quantify the level of melanism in these various species. So obviously I went out and I sampled lizard populations on the lava flows, and then most importantly, for every one population on the lava flow, I sampled 2 non-lava flow populations. And that I used this kind of population trio approach, not only for the phenotypic data that you see here, but also for the genotypic data that I'll show you later. And that allowed me to not only compare lava flow to non-lava flow, but to determine whether the variation that we see between lava flow and non-lava flow is within the range of natural variation that we just see between populations in the normal desert. And here's what I found. So we measured luminance, which is essentially brightness. Darker individuals have lower values here. Brighter individuals have higher values. And for 2 non-lava flow populations, and then 1 lava flow population. And these are the data for Crotophytus. So as you can see here, the Crotophytus from the Carrizoso lava flow are significantly darker than the 2 non-lava flow populations, but at Pedro Armendariz, they're not significantly different than the non-lava flow populations. They're not significantly darker. Scoloporus shows the same pattern. Again, at Carrizoso, they're significantly darker than the non-lava flow populations, but at Pedro Armendariz, they're indistinguishable in terms of brightness from the non-lava flow populations. And so that was surprising. Urosaurus shows a similar pattern. Again, from that middle-aged Aden-Afton lava flow, they are significantly darker than the 2 non-lava flow populations that I measured. And so, in conclusion, for question 1, the lizards from Aden-Afton lava flow and Carrizoso, kind of regardless of species that I looked at, were melanistic. However, lizards from the Pedro Armendariz were not. And so that kind of begged the question to me, what's going on with the Pedro Armendariz lava flow? And a hunch that I investigated came from just observing these lizards once I had captured them. So I noticed that these lizards change their coloration pretty rapidly over short time scales. So this is the same lizard photographed at very well below active body temperature and then at active body temperature. And you can see this lizard darkens and lightens pretty dramatically over a course of a few minutes. And this is something that's well known. Melanosomes, which contain little granules of melanin, will disperse those melanin granules to absorb light more readily, or bring them in and concentrate the melanin granules to be able to reflect. And so this occurs with temperature, but kind of begged the question of whether plasticity might play a role in explaining the difference in coloration that we were seeing between Pedro Armendariz and the Carrizoso lava flow. So I asked the kind of just general question of how variable melanism was over time. And by over time, I mean over short time scales here. And so in order to do this, I zoomed in on just one species so I could really increase my sample size and focused on on Crotophytus from the Pedro Armendariz lava flow and the Carrizoso lava flow. And I'll show you the results of that now. So here's again the same graph that we had seen earlier. Again, these are samples taken at active body temperature when these lizards are probably at the temperature that they maintain throughout the majority of their day. And again, the Carrizoso lava flow is significantly different from the non-lava flow populations, but the Pedro Armendariz individuals are not. So what happens when you quantify the brightness of those individuals at colder temperatures? Well, you can see that Pedro Armendariz lizards start out pretty similar to the Carrizoso lava flow lizards, but and that is much darker than the non-lava flow lizards get, but they actually change significantly in coloration from cold to warm, and unlike all of the other species that remain, all of the other populations which remain pretty constant in their coloration. So the Pedro Armendariz lizards can change color rapidly and significantly, and they have the kind of widest potential range of brightness as well. And so, if you'll bear with me, we'll get a little hand wavy here, but I think this is really interesting. I think it may show that plasticity may play a role at Pedro Armendariz. And obviously this is only one aspect of plasticity. We didn't investigate others. This is just physiological color change. But it's possible, again, I have no evidence of this, it's possible that this could be an adaptation to the more heterogeneous environments, heterogeneous backgrounds that occur at the Pedro Armendariz and not at the Carrizoso lava flow. So that brings us to the third question, which is a much more general question, but has really interesting applications that I will explain. And so I asked whether the lava flows were barriers to gene flow. And so I just showed you that some of the populations that I studied are indeed phenotypically divergent. They are significantly darker than the surrounding non-lava flow populations. And given that lizards are mate through visual cues, you might assume that phenotypic divergence could lead to reproductive isolation. If that coloration is important for mating, it could lead to reproductive isolation. And then I would say as well, this question would get at kind of the demography and natural selection modulators that I talked about earlier. The demography maybe most obviously because we're actually explicitly measuring how much migration would occur between lava flows and non-lava flows, and then natural selection I'll talk about a little bit later. And so this general form of question, which is inherently a pattern-based question rather than a process-based question, is one that I got from the isolation by environment literature. And so just like lizards on lava flows, you can imagine that 2 green lizards are adapted to live in these green trees and 2 blue lizards are adapted to live in these blue trees. And we're just measuring the level of genetic discontinuity between those green and blue lizards. And that's kind of agnostic of the process that might be generating that. Right? The lizard, there could be strong natural selection against immigrants. There could be preferential mating to mate with similar colored lizards. There could be some kind of hybrid inviability. That all of those could generate the pattern that we're observing here, but the technique that I'm using is kind of agnostic of that. We're just measuring the pattern. And so we do this in a really simple way. What we'll do is, what I did was to plot the genetic distance between individuals against the pairwise geographic distance of those individuals. And you would expect that 2 individuals that are further apart would be more genetically distinct than 2 individuals that are closer together. And so we'd have this increasing genetic distance with geographic distance, a pattern of isolation by distance. However, if you color those comparisons by whether they're on the lava flow versus off the lava flow, or on the green trees versus on the blue trees, whenever you compared between different environments, you would if there was an effect of the environment, you would expect those different comparisons to cluster anomalously higher given some geographic distance. And so that would be evidence for isolation by environment or the environment being a barrier to gene flow. So this is what I did. I created a RADseq library with 96 individuals for all 3 species. I ran it twice on an Illumina high-seq lane and got thousands of SNPs putatively from across the genome at pretty high coverage. This was a data set that I had pretty high confidence in. And here are those actual graphs. So here, this is pairwise pi, a pairwise genetic measure of genetic distance between 2 individuals. Each one of these dots is a pairwise comparison. And on the x-axis, pairwise geographic distance. And for this graph here shows both lava flow populations and all 4 non-lava flow populations. And you can see that these comparisons between lava flow and non-lava flow individuals, these red dots don't cluster significantly higher than we would expect under a pattern of isolation by distance. So while isolation by distance does appear to occur, isolation by environment does not. And the same pattern holds for Scaloporus. Again, this is both lava flows and all 4 non-lava flow populations grouped together, and we don't see the pattern of isolation by environment. We don't see those red dots clustering anomalously higher given some genetic distance due to isolation by distance. And the same pattern also holds for Urosaurus. And so one explanation for this is that ongoing migration may be kind of homogenizing the genetic differences that we would expect to see among these lizards. So we actually explicitly quantified migration using Dan Portig's DADDY pipeline and found that in all comparisons, indeed, ongoing migration did occur, and I should back up and say that models that included migration better explained the data than models without migration, and migration was ongoing in all of these lava flow, non-lava flow comparisons. So are the lava flows a barrier to gene flow? The answer is no. We see phenotypic differentiation, but not genome-wide differentiation. And I should say we see phenotypic differentiation in only some of the populations, but not genome-wide differentiation. And that pattern doesn't really matter whether you have strong phenotypic differentiation or not. And we find evidence for ongoing migration. And so to kind of wrap things up regarding the lava flow lizards and about those modulators of convergent evolution that I spoke about earlier, it appears that we do have convergent phenotypes at some lava flows, but interestingly, a similar pattern of demography at all the lava flows. And so that's interesting. They converge at the level of phenotype, but the pattern of demography occurs similarly across all lava flows. And unlike, as someone might predict, there's no buildup or erosion of reproductive isolation with that phenotypic differentiation, right? With the lava flows where lizards are really dark and really distinct, there's still no reproductive isolation, and it's very similar to where the lizards are not distinguishable from the non-lava flow populations. And so in at least Caryzoso and the Aden-Afton lava flow, this is a cool example of potentially a selection-migration balance. Again, we haven't measured natural selection, but if you take the level of background matching or this convergent evolution as evidence for natural selection, this could be a cool selection-migration balance where those melanistic phenotypes are persisting in the face of strong ongoing migration. You can also look at this as just snapshots in the life of the lava flow. And so this gets at kind of the time aspect of that modulator. So when lava flows are 5,000 years old, lizards tend to be homogeneously dark and significantly darker in coloration. Same with a little bit of heterogeneity, but something happens in between here where that degree of melanism tends to disappear, even though regardless of age of the lava flow, migration is ongoing in all 3 of these populations. And so there are a lot of questions that are still remaining that I'm really interested in answering about these lava flow lizards. First of all, as I mentioned, or as I alluded to earlier, interesting questions about the ecological aspects of the evolution of melanism. Why was it these certain these certain species, which are only a subset of the total desert fauna that could potentially colonize these lava flows? You can actually ask cool questions about the genetic architecture of adaptation. So there's good evidence that melanism is a genetically based trait, and that, Bree and Ammon and others have found that that melanism does not change ontogenetically, that melanism is indeed heritable in at least other lava flow lizards, and we know from a variety of other species that there are genes and mutations that can directly correlate with melanism. So there's good evidence that this is I would say I would put my money on this being a genetically based trait. And one really simple explanation of why we didn't see a genome-wide pattern of differentiation is that there may be one or a few loci that are under selection, and so you might only expect kind of those genomic islands of differentiation rather than a genome-wide pattern of differentiation. And this is also another really interesting place to study a kind of classic question of whether these adaptations arose de novo in these lava flows, or given that there's ongoing migration, whether they arose once in kind of the oldest lava flow and then through introgression have made their way to to the younger lava flows. So whether this adaptation arose from standing genetic variation or de novo. So now we'll take a kind of big shift and talk a little bit about voles. So this is the Amargosa vole, and I really wanted to, as Bree said, apply the techniques that I had been using to study evolution and apply them to something that could really do good in the world and help inform conservation. So I asked the general question of just how population genetics can inform conservation, but I think we all know the answer to that. The answer is in many ways. I'll give you a really concrete example of how that happens. So the California voles are Amargosa voles are a subspecies of the California vole. There are 17 described subspecies, and they range kind of throughout the California floristic province. Recent work by Chris has shown that they do have a strong break, a strong north-south break that basically corresponds to the transverse range in California. But depending on which loci you use, you get a kind of different answer regarding the placement of the Amargosa voles. So the Amargosa voles are found here in this small little patch out in the Mojave Desert, and using mitochondrial DNA at least, they fall out in the northern clade. I should also note that for your interest, these two mitochondrial clades don't correspond well to the 17 described subspecies of voles. The Amargosa voles are limited to about 10 square kilometers around the town of Tecopa, California, where springs or water come to the surface and form this mesic vegetation that they really depend on. Their habitats are threatened by climate change and drought, but also off-highway vehicle use, overgrazing, and draining for agriculture. And so the Amargosa voles were originally described in 1891 from a town north of Tecopa called Shoshone, and it wasn't until the 1930s that actually our very own Annie Alexander actually rediscovered these voles in Tecopa, California. They were listed as an endangered species pretty much as soon as the Endangered Species Act came out, but real conservation work didn't begin on them in earnest until about 2008. Around then, they censused the population, and it was found that only 140 to 240 of these voles remained in the wild. So this is a really imperiled species. So I was called in after a really dire situation arose in 2014. And so what you can see here is a satellite image of Marsh 1. This was the marsh that contained the largest number of voles in the Amargosa voles in the world. And you can see what happens to this marsh as we go forward in time from 2009 to 2014. And that corresponded with not only construction at this underroad culvert here that forced the marsh to drain, but also one of the worst droughts in California's history. And so let's see if it'll play. Yeah, cool. So you can see the marsh goes from having this really dark green, good mesic habitat for voles to just completely draining into here and then drying up with the drought. All of these pictures were taken in May. And so at this point, when managers noticed this kind of catastrophic decline of voles, of the healthiest population of Amargosa voles, they took 20 of the individuals, brought them back to a captive colony at UC Davis for hopes of not only creating an assurance colony, but also reintroducing those voles back into the wild. And I was called in to kind of do the genetics on the situation. But I was really interested to see how population genetics, the tools that I was using before, could affect conservation. We know the answers, but here are 3 explicit ways that population genetics can affect conservation. So one, we can just resolve that question of who the Amargosa voles are most closely related to. We can determine levels of genetic diversity in the wild, and then determine how much of that genetic diversity was captured by the captive colony, and thus inform not only captive colony management, but the reintroductions that occur later. So that's exactly what I did. I used best rad approach, another RADseq approach, sequenced 92 voles, the majority of which were Amargosa voles, both from the wild and from the captive colony, but that also included 28 desert-dwelling California voles, so we could kind of compare levels of genetic diversity that we saw in the Amargosa voles to what we might expect from a more typical population of desert-dwelling voles. We got thousands of loci, again, putatively across the genome of these voles at reasonably high coverage. Again, a data set that I'm pretty proud of. First, I ran a space mix analysis to kind of get an idea of the phylogeography. And so here you can see the dots here represent the actual geographic sampling locations of these voles, and each one of these ellipses is a 95% confidence interval of the geogenetic location. And so individuals are pulled to their geogenetic location depending on the amount of admixture that they share with other voles. And so to make a long story short, the Amargosa voles are pulled towards this kind of common nexus of northern clade voles, indicating that they share more admixture with the northern clade than the southern clade. And I also created a tree from the SNPs that I got from the RADseq, and so using snap tree, the Amargosa voles also fell out nested within the northern clade, indicating that they're probably nested within the northern clade rather than the southern clade. The southern clade actually came out sister to the prairie vole, the outgroup. Measuring the genetic diversity of the wild Amargosa voles, I found that Amargosa voles have about half the genetic diversity as those other California, desert-dwelling California vole populations, and were an order of magnitude more inbred on average than the desert-dwelling California voles. Looking at these data in a different way, we can also see that there's significant population structure, that the wild Amargosa voles, even though they have so little genetic variation remaining, it's significantly structured into 2 distinct groups. And so we can look at this as an admixture plot, and we see that some marshes are made up of pretty much only kind of green group alleles, and some almost completely of orange group, but there is actually a lot of admixture among these 2 groups. And really, the difference between these 2 groups is only equivalent to an FST of 0.08. So this is really kind of shallow population structure, but it is significant. If we add in the captive voles, we can see that maybe unsurprisingly, they all fall out within this green group, which, if you remember, is the group that that Marsh 1 originated, or that Marsh 1 was a part of, and that was the marsh that they all originated from. And so the captive voles don't really capture the genetic diversity of this orange group. And looking at this another way, we see that it makes sense that the captive voles have about half the genetic diversity as the wild Amargosa voles, but interestingly, they're actually less related to each other than the wild Amargosa voles on average, which may be evidence of really good captive breeding procedures in the captive colony. And so this brings us to the conservation and management implications of this work. Now we know with pretty good certainty that if the Amargosa voles go extinct and reintroductions are warranted and approved, we would want to replace the Amargosa voles with northern clade voles. Those are the voles that they're most closely related to. In terms of captive reintroductions, we have, and translocations, we kind of have two choices. We could preserve the genetic structure, right? Like mate orange voles, or reintroduce orange voles to orange marshes, reintroduce green voles to green marshes, or we could kind of translocate and reintroduce voles to any suitable habitat around Tecopa, regardless of the genetic structure. And so, given that the structure that we observe is relatively weak, an FST value of 0.08, given that these marshes can kind of blip in and out of existence, and we know that California voles too experience really large population booms and busts, I argued that we should translocate and reintroduce Amargosa voles to suitable marshes regardless of their genetic structure. And I should also say, like, given that these voles exist over an area of 10 square kilometers, outbreeding depression might not be something we would actually be worried about. So that's even more evidence for kind of spreading all these metaphoric eggs into as many metaphoric baskets as possible. And actually, this is what has begun to occur now. So voles from Marsh 1 are being translocated and moved into other suitable habitat areas, and most of the marshes that have voles in them are now at carrying capacity. So it's working out well and will continue into the future. And so this was a really cool example, an example that I was really proud of where I was able to use this kind of esoteric genetic analysis that I use to study convergent evolution and evolutionary biology and apply it directly to have some effect on the conservation management of an endangered species. This was something I was really proud of, and yeah, we'll see how this goes forward in the future. So now we'll kind of zoom out a little bit. I'll give you some conclusions from 5.5 years in grad school. And a lot of this will be kind of old news for the old hats here in the audience, but I think bears repeating for the grad students in the audience. As evidenced by my lava flow lizard study, each study, I ended that study with more questions than I actually began. And I think you should expect that through your PhD. As Perry de Valpine taught me, and I'm proud to admit he was right, you should plan your stats before you begin. Like, create your experiment with the statistical analysis that you will use in mind. And then, more importantly, I think, do your power analyses in the field. Once you're able to determine what an effect size would be for your study, do your power analyses well so you can know when you have the right sampling, enough samples, I should say. And then finally, something that is that everyone here knows well, but really informed my work in my PhD, you should observe, learn, and treasure the natural history of your organisms. That really informed some of the work that I did, and I think is crucially important. All right, so on to acknowledgements. Obviously, I'll acknowledge Bree. Bree has supported me throughout my scientific career and throughout my personal development, my kind of happiness here in grad school, and that was everything I could have hoped for. It was really awesome to know that you always had my back, Bree. So thank you. The Rosenblum Lab were my best friends throughout this entire time, and we've done science together. We've gone in the field together. I literally would not have been able to do this without the Rosenblum Lab. They're my best friends, and they're great. Throughout the work that I've done, throughout a lot of the genetic analyses, I spent a lot of time searching for the best K. But little did I know he was here all along. KB was absolutely responsible for making me the bioinformatician that I am today, and I think he's as valuable as some of the specimens here in the lab, or here in the museum for his expertise. So thank you, Kay. And then I'd like to acknowledge my other friends and family, my field assistants, all the people that gave me permission to go on their land, the DeWoody Lab and the Foley Lab for collaborating, all the people that gave me funding, including and especially the MVZ, and then all of the lizards and voles who I wouldn't have been able to do this dissertation without. With that, I will take questions. Land, so BLM is there. UC Davis is actually running the captive colony and the reintroductions. And Cal Fish and Wildlife is also participating as well. So it was really interesting to talk with all those diverse managers who all had kind of different perspectives on what genetic answers they wanted, and kind of having to navigate that and design a study that fulfilled all of their desires.
Apparently they were listening to you.
Yeah, exactly. I was listened to, which was very cool as well. Yeah. Yeah. Yep.
It's interesting that you don't see any evidence for isolation by environment. I was thinking it seems like maybe a good idea to formally at some point rule out the possibility that it could be developmental plasticity. So I'm curious what your thoughts are on that, or if you could elaborate on that. And then also to ask whether you see any individual loci that are FST outliers or that show evidence of isolation by environment that might be linked to the alleles that are under selection. Yeah.
So regarding plasticity, I didn't do any of those analyses. I actually wanted to. I had big plans to go and collect gravid females from the wild and bring them back, but turned out that my field season coincided with a really strong drought in the Chihuahuan Desert, and of 80-something lizards, male and female, that I caught, only one was actually gravid that summer. And so, however, other people have done that work explicitly with Euta stansburiana on a California lava flow and with Scoloporus on the Carrizosa lava flow, and found that juvenile coloration correlates very strongly with maternal coloration, and that coloration doesn't change ontogenetically regardless of what habitat you're raised on. So regardless of whether you're raised on black lava sand or even white sand. And so that's not, I can't say that for sure. I can't rule developmental plasticity out for all of these lizards, but given that where we've looked for it, it doesn't occur, and given that there are a lot of examples of a genetic basis of melanism, I would put my money on it being genetically based. In terms of FST outliers, I have done some FST outlier tests, and surprisingly haven't found any significant outliers. I haven't used all the programs. I used 2, and that is something I really want to look at in the future, especially given that there's very little population structure among lava flow and non-lava flow populations. It'd be if there were FST outliers, you'd have a really good chance of detecting them. So yeah, that's definitely something I want to look into in the future. Yeah.
Just to follow up on that, do you think of maybe using annotated genes in order to find those functional sort of alleles that you're looking for, since the SNPs weren't as effective? And then secondly, does that kind of have implications of talking about selection on the gene level rather than the organism?
Yeah. Yes, so I have tried kind of vaguely to align the RADseq data that I have back to the Anolis genome, which is pretty well annotated, but haven't had much luck with that. And so it wasn't, so what I was kind of waiting for was to find genes that or at least SNPs that segregated, that were really divergent, and then seeing if those specific regions would map back to the Anolis genome, but haven't had much luck there. And then looking at the level of the gene, yeah, I think there's a lot of explanations for why we didn't find a genome-wide pattern of differentiation. And I think one thing that's totally possible is that given that even though I have thousands of markers putatively across the genome, that still only represents 2 or 3% of this entire genome. So there's a significant likelihood that I just missed either those specific genes or even areas of the genome linked to those specific genes in this RADseq analysis. And yeah, so that is possible. Yeah, Jim?
So one of the premises, like the fundamental premise, I think, of the Chrotophytus work is that the selective pressure on these Chrotophytus, on the coloration of the Chrotophytus, is background matching. But you know that in other Chrotophytus species, like in Chrotophytus dickersoni and Chrotophytus nebrius, and even in other populations of Calaris, they can be the males can be really vividly colored. They can be bright blue, bright green with yellow heads, that sort of thing, and they don't seem to be trying to match their rocks at all. So I'm just wondering, but the females tend to be more drab. So I'm wondering if you saw. I have two questions. Based on that premise. The first is whether or not you looked at females separately from males, and whether or not they might show more of the pattern you would expect. And the second is whether, if you would care to speculate on why some Chrotophytus populations might actually be background matching and other populations wouldn't be.
Yeah, sure. So yes, we definitely looked at the effect of sex on the findings that we had here, and most importantly, we were here at kind of the height of the breeding season, so you would expect the males to be as different from the females as you would ever expect, and we found no effect. Males and females were slightly different, but not significantly different in both how dark they were and how well they matched their substrate. And so in terms of speculating, I really didn't have a good answer for that. One thought that that occurred to me is that the non-lava flow rocks that these Chrotophytus are matching to are very, very far away from even their normal kind of typical coloration. And so it might just not be possible for a Chrotophytus to background match to that. And they might have some other adaptive strategies, maybe behavioral, that would allow them to effectively avoid predators in those areas that weren't camouflaged. Whereas the lava flow rocks are actually not that different from the coloration of even in terms of brightness, I should say, of even the non-lava flow populations. And so maybe their crypsis would be more useful, and there might be a selection for crypsis at those locations.
Easier to get there.
Right, exactly. I don't have any evidence of that, but that would be something I would be interested in looking at in the future.
Do you know anything about the diet? Because there is, people have proposed that in Chrotophytus calaris specifically, that vibrantly colored populations tend to be populations that specialize on eating insects, where background matching probably doesn't matter as much, and the more lizard specialists.
Right, yeah, and I don't have any data on these populations here, but just in terms of lizards encountered over person hours used, there's a much higher density of all lizard species on these lava flows, so it would make sense that their diet could shift to a more lizard aspect. Yeah.
Alex, you described this plasticity on the Armendaris lava flow and speculated that it might be adaptive plasticity. Are the lizards associating with dark rocks when they're cooler, and are they using changes in temperature to background match?
I did not find any evidence of that. That I didn't explicitly measure that, but pretty much all of the lizards, regardless of their temperature, were found on the dark rocks. And so I'm not, that's why I was very hand-wavy, because I didn't explicitly measure other aspects of plasticity, and it seems like even though the background is more heterogeneous, you still only find lizards on those black rocks, and so I think the picture is pretty complex.
So when they heat up, they don't match as well? Is that
correct, yeah, they do. They contrast more, and they're definitely easier to spot, but you still find them on those black rocks. Kind of going back to what I was saying with Jim, I bet that there's a behavioral trade-off as well, that they're more wary and more eager to flee when they're warm on the Pedro Armendaris lava flow than possibly on the Carrizoso. Yeah, in the back.
I'm sure you know, but in Bio 1B, there's a lab about mice pelage on the same lava flow, at least in Carrizoso. I'm wondering if you could hand wave some comparisons maybe between these two, the basis of those studies and yours.
Yeah, so the mice at Pedro Armendaris are indeed significantly melanistic. And so that kind of goes against the pattern that I was finding here in lizards, and also is interesting. I mean, a lot of them, well, the mice that you study in Bio 1b are actually nocturnal, and so they're facing a different suite of predators, even though they're both probably raptors, than these than the lava flow lizards here. And so yeah, that is actually surprising, because those do display striking patterns of melanism, whereas these lizards technically do not. Yeah.
I was wondering, when you were sampling at these lava flows, did you ever do like a transect from the lava flow off and kind of see if there was any sort of relatively dip in densities going off the lava flow?
Oh, yeah. So I did notice pretty immediately that, so I should say that these lizards are not like kind of continuously distributed throughout the habitat, but yeah, pretty much some of the non-lava flow populations were like 1 to 1.5 kilometers away from the actual lava flow, and even there were still a lot harder to find. It took more person hours per lizard at those non-lava flow habitats than elsewhere.
Okay, well, let's thank Alex for a great talk.